So I'm going to tell you about a story about two libraries, and technology does make a difference. How do I forward this? So this is Octopus Hot Spring, and this is Tom Schoenfeld tending his gear, watching to make sure none of the tubes fly apart. And he had an original idea a number of years ago that if you want a better polymerase, you should look for a viral, true replicating polymerase instead of a DNA repair enzyme. And the thinking was that perhaps a thermostable viral polymerase would impart new properties to take advantage of in various applications. Can I have the next slide, please? Oh. Oops, sorry. So this is the process that we go about doing it. You have to have a permit, take a bunch of gear out, filter, boiling hot water, and convert it into DNA. This is before Feed29 was around and before next-gen sequencing was around, so we had to figure out a way for anonymous amplification and cloning, and then JGI, thank God for JGI, sequenced about 10,000 clones from both directions, so we got about 20 million reads. And out of that process, we found a really interesting polymerase. And it was the only one that expressed very well, and it had a bunch of novel, unique properties. One of them is that it's a true reverse transcriptase enzyme compared to TTH. You can see it does a much better job at RT-PCR. It also has fairly high fidelity and some other interesting properties. It does PCR, as I mentioned, but it's still not a perfect enzyme. There's issues with it. Didn't work for everything. But one of the other unique properties that we found that it had was strand displacement. So I believe this is the first example of a strand displacing RT polymerase that you can use in a RT lamp assay. And what's really nice about this technology is it allows you to, within 30 minutes with dirty samples, detect what your pathogen is if you have the right primers. And in this case, some of the team members developed an Ebola assay that went over to Africa with Tony Goldberg from the University of Wisconsin, and he was sampling rodents and other critters out in the bush to see if they had any traces of Ebola around. And right at the time they did this assay, this guy decided to sit on the fence post. So that was the story of the polymerase. We were very curious as to whether there was more to the virus story. And so we made a second library. This time we used Feed29 DNA polymerase, rolling circle amplification. We got a whole different set of results. The first library we had 20 million reads, but only 3 of them were that we could tell were to this virus polymerase. In this case, you can see quite a few of the reads mapped to this one contig. We call it Octopus Spring 3173 virus. And it's a pretty clean library, only a couple % of bacterial contamination. We also assembled a Pyrobaculum spherical virus out of this library. And this is supposedly not been published, but it's supposedly old news to people in the know. There is quite a good bit of Nano Archaea, Archaea bacteria present in this alkaline hot spring. This is the pink filament Aquifocales that we believe is the host. So we were able to, at very high stringency, able to assemble a complete circular genome, 37 kilobases, and it turns out it has quite a few annotated features on it. There's a bipartite replisome-like set of genes going counterclockwise, and the more structural things going clockwise. Here's kind of a synopsis. There's 48 genes total, annotated 17, and hypothetical 10 and unknown. There's an interesting story about the diversity. So this is a map of the genome, and you can see some of the labeled genes here, the annotated ones, and you can see the the non-synonymous amino acid changes are very low in certain genes, like this little capsid guy, but much higher in other places. Extremely high for this unknown, unannotated gene. I find it kind of interesting. This is the DNA polymerase gene. It's a polyprotein, which is highly unusual for DNA viruses. It's got a helicase-like annotation at the amino terminus, and the last 600 amino acids or so is a polymerase. Far fewer changes tolerated in that region. There's some structural variations in this library, so if you lower the stringency, you can assemble things that are coming in and out, like this methyltransferase. This is not an artifact of assembly or sequencing. We actually cloned and sequenced some things and found this exact thing in a clone as well. So to my surprise, once we had this polymerase gene, it was interesting to see if there was other related genes out there in the databases, and only in the JGI database will you find these. And it turns out we've got cousins to Yellowstone National Park, alkali-like hot springs, octopus, conch, bath, and so on, and then a couple of Great Basin hot springs like Little Hawk Creek and Great Bowling Spring. And here shows the kind of features. They're all fairly high temperature. There's one that's a little low. They're almost all alkaline except for that one. And the identity that we blasted to is fairly decent. But to our surprise, the hits contain, you can see there's, here's the number of hits, but there's some very large scaffolds that came out of this from the JGI database. And this is the cousins, and this is the original circular genome that we deduced from our library. And you can see the synteny is very high amongst these other derivatives from these other hot springs. And you can see the orientation of the replosome versus the structural proteins. And what's interesting is if you look at the annotated features, you can see there's some core phage capsids and terminases and the polymerases. And there's always a nuclease and a helicase right in front of the polymerase in this order. And in one case, it's reversed. So there's annotated genes that are coming in and going away. So it's an interesting story from what's the ecology of these viruses, what are they doing. The other interesting feature is that in a couple of these, the polyprotein is split. It's no longer a polyprotein. The polymerase domain is not fused to the helicase domain. So what is the host for this metavirus? And it turns out the very same year we collected our second sample to make this library, another group collected pink filaments from octopus and sequenced it, and it turns out there is a familial host here. So we blasted the entire octopus circular virus against the CRISPR finder database and found these hits. And this shows the spacers and their identities. And when you go into the JGI database and blast it, you find these contigs that are associated with this. And then if you blast that to the thermochrinus genome, which was cultivated from octopus 20 years ago, you see a lot of identity. Ironically enough, there are no protospacers for our virus in this isolate from octopus hot springs from 20 years ago. So I'm going to switch back to the polymerase and tell you one more little vignette. It's kind of a fun story. We don't have a crystal structure. It's very distantly related to other polymerases, surprisingly enough, to a apoplast from Plasmodium falciparum. There's some identity there. But we were interested in trying to improve the enzyme, fix some of its flaws. So we did some design of experiment mutagenesis with Tom here in the Bay Area. And one of our mutants, surprisingly, had a amino acid change that knocked out the RT part of the enzyme, but not the PCR. So here's the wild type and one mutant and the one that knocks out the RT. So both enzymes will do PCR. One of them won't work on an RNA template versus the other one will, which is kind of surprising. So we mapped it out. This shows Taq DNA polymerase around the motif A, which is located within the palm subdomain. It's also where the incoming nucleotides are incorporated. And it turns out there's some of these mutations in Pol A that make it a weak but observable RT. And one of our mutations to that mutation abolishes it. So this is a nice, interesting example of a single gene, single amino acid change abolishing an activity, but not the DNA version of that activity. have had a lot of collaborators over the years, and want to thank them, Brian Hedlund and Eric Womacker here, and especially Tom Schoenfeld, who had a lot of good ideas and showed really excellent scientific leadership in this whole project. That's it. Thanks.